Okay, welcome to this session on the presentation of a new MSc Neuroscience at Cardiff University. The way we are planning this session is that you will have three short presentations, one by myself, Dr. Xavier Caseras. I'm the lead of the program, and I will introduce you the program in itself. And then two of my colleagues, Dr. Rebecca Sims from the Dementia Research Institute will give you a short talk on the research that they do at the Dementia Research Institute, and it will be a bit of a taster of the type of things that you will learn if you were to enroll in this MSc Neurosciences. And then Professor Anne Rosser will do the same with the research that they do in the Advanced Neurotherapies Center. So you will have a presentation on the program itself, and then two short lectures or two short talks that will give you a bit of a flavor of the type of things that you will be learning if you were to enroll in our MSc. So without more further ado, I'll share my screen and I'll take you through my presentation. So this is what we are presenting today is an MSc Neuroscience, and this is an initiative led by the College of Biological and Life Sciences at Cardiff University, and it's a program that is participated by the Schools of Medicine, the School of Biosciences, and the School of Psychology. And why these three schools have joined forces to prepare this program and deliver this program will become obvious in a minute. It's a one-year duration course, full-time, delivered face-to-face at Cardiff University, and is research-led teaching. And the aim of our program is to provide the students with the knowledge and practical skills to advance the neurobiological mechanistic understanding of neuropsychiatric conditions, and to translate this understanding into better diagnostics, treatments, and clinical management of these conditions. So despite the generic title of MSc Neurosciences, it is a program with a specific focus on neuropsychiatric condition with an important clinical perspective in it, but it also intends to provide you with knowledge and skills to apply that knowledge into the bedside or into clinical practice. Why an MSc Neuroscience at Cardiff University, and why an MSc in Neuroscience delivered by three schools? Neurosciences is obviously a multidisciplinary field, and in Cardiff University we take that very seriously, and we've been working together, the School of Medicine, specifically the Division of Psychological Medicine and Clinical Neurosciences, and the School of Psychology and the School of Biosciences. We've been working collaboratively for many years now in advancing our knowledge and applications in neuroscience. And you see here in this graph the three schools represented in this three-dimensional space, and you can see in the center the Neuroscience and Mental Health Innovation Institute, created many years ago at Cardiff University collaboratively by the three schools, that actually tries to push research into neuroscience and mental health by also with the stakeholders and industry try to find applications to the research findings that collaboratively the three schools develop. You also see there that there is another institute, the Hodge Center for Neuropsychiatric Immunology, that does similar things that the Neuroscience and Mental Health Innovation Institute, but more focused in the crossover in between immunology and neurosciences. You also see that we have developed other institutes and centers collaboratively in between medic and biosciences, the Dementia Research Institute that we've mentioned before and that you will hear more about from Dr. Rebecca Sims, the Advanced Neurotherapy Center that tries to deliver new neurotherapies and new methods of tackling mental health or neuropsychiatric conditions, and you will hear as well more about it from Professor Anne Rosser later on. The Medicines Discovery Institute that takes research and findings from biosciences and medical, also psychology, and tries to identify new targets and develop new interventions, new medicines for neuropsychiatric conditions. We obviously have Cardiff University Brain Research Imaging Center, a very advanced neuroimaging center with fantastic brain imaging, in vivo human brain imaging technology that is participated by psychology and the Division of Psychological Medicine and Clinical Neurosciences, that is pushing the boundaries of human brain imaging and applying these methods to understand better neuropsychiatric conditions. Then we have the Center for Neuropsychiatric Genetics and Genomics, the Wolfson Center for young people mental health, the Welsh Autism Research Centre. So a large range of institutes and centres that have actually been created collaboratively between the Division of Psychological Medicine and Clinical Neurosciences, the School of Psychology and the School of Biosciences, that work specifically in neuropsychiatric conditions and neurosciences, and therefore a long history of inter-school collaboration that has now come into fruition into these MSc Neurosciences that we are presenting today. And what type of work do we do here at Cardiff University? You might be asking. Well, to give you a bit of a flavor of how we navigate our understanding of neuropsychiatric conditions and how this cross-school collaboration brings results. In the Center of Neuropsychiatric Genetics and Genomics, we are leading international consortia and international research trying to identify, for example, common and rare risk variants that predispose or increase your risk for mental disorders. But in collaboration with our colleagues in psychology and in Kubrick, in the Center for Neuroimaging, what we also do is to actually investigate what genetic predispositions or which genetic variants might actually modulate how brain develops and how brain morphs or changes and how brain functions, and what type of genes affect the functioning and the anatomy of the brain. And obviously what we then do is try to actually look at the interconnection or the overlapping between genetic risk for mental disorders and genetic variants that shape the function and the anatomy of the brain, and try to find commonalities in between these two, and therefore try to identify pathways that help us to determine the neurobiology behind neuropsychiatric conditions. We also take these genetic findings that we have, and we take it in collaboration with biosciences and the Medicine Discovery Institute, and we try to identify new targets to develop new intervention, new pharmacological interventions for mental disorders, but also to work as you can see here in this example. What we also do is to try to investigate how current medications can actually be absorbed differently or have different efficacy depending on your genetic component or your genetic makeup. So how different individual differences in genetics might actually result in different effects of existing medical interventions or pharmacological interventions. We also take, in collaboration with our colleagues in biosciences, we take these genetic findings into the lab, and then and you will hear more about it from Anne Russell later on, we develop neurons and we grow neurons from stem cells. And we also develop animal models, and then we can more specifically look at how neurons communicate, how they grow, how they myelinate. So we can look at the anatomy and the function of specific neurons or organoids into the lab, so that we better understand how genetics can affect electrophysiology, for example, or the communication in between specific neurons that have been grown for having specific genetic characteristics. With our colleagues in Kubrick, we work very much in developing new methodologies, new methods of looking at in vivo human brain, getting to the level of almost histological resolution. We can look at dendritic arborization within grey matter. We can actually dissect white matter tracks and look at the specific components like myelin content or axon caliber. So we can really look at very detailed resolution of brain anatomy and brain function, and then we use that knowledge to try to compare in between different types of psychopathology and neuropsychiatric conditions and large groups of healthy controls, trying to identify biomarkers that might be definitory of certain neuropsychiatric conditions. We take that knowledge then to look at how certain genetic variants might explain those changes that we see in patients in the brain of people suffering certain neuropsychiatric conditions. We, as we said before, we investigate which genetic variants might actually be modulating those responses or changing or producing changes in brain anatomy and function, and all that with obviously the final intention or goal of bringing that knowledge and those advances back into the clinic. And in here you have a really novel and good example of that, which is this new therapy, gene therapy that Cardiff University has been developing in Huntington's disease, reimplanting genetically modified neurons within the brain. And you will hear more about it from Anne Rosa later on. So basically, we are pioneering in neurosciences. We develop different methodologies, and we have expertise in many areas related with neuropsychiatric and the applications of those neuropsychiatric advances into the treatment and understanding of the pathology in neuropsychiatric conditions. So how the course look like, or how we are going to try to transmit all that knowledge and practical skills within the MSc Neurosciences. The delivery of our teaching is divided in blocks of 4 weeks or 7 weeks. And within each of these blocks, you would be taking 1 or 2 of these modules. So in green, you have core modules. This is elements that we consider to be core to our program. And in blue, you have the optional modules. So the program starts with a block of 4 weeks in which you will be introduced into neuroscience, and you will be taught about all the fundamental knowledge that you will require to then progress successfully for the rest of the program. These 4 weeks, you will learn about brain anatomy, neuronal communication, basic genetics, but you also will acquire basic skills that you will require for the rest of the program, like, for example, how to do a review of the literature, how to report a review of the literature as well. You will learn about reproducible science. You will learn about statistics and the use of the scripts and the use of R to perform these statistics. So not only knowledge, but also practical skills that you will require for the rest of the program. Then you will dive into another block of 7 weeks in which you will learn about the methods of in vivo human imaging and disease modeling in the lab. These modules as well will include theoretical knowledge, but they will also include practical sessions in which you will learn how to deal with imaging data and data from lab models of neuropsychiatric conditions, and you will learn how to analyze and how to extract knowledge from these data sets. Then the following 7-week block, you will be engaged in another core module, the one about genomics, again, including theoretical knowledge, but also practical skills. And then if you decide so, you would also be engaged in one of our optional modules, which is about interventions in brain disorders. And in this module, what you will learn is how these 3 techniques, how disease modeling, in vivo human imaging, and genomics can actually be used to develop new interventions in neuropsychiatric disorders, how we use that to develop new interventions in psychiatric disorders. Of these 3 optional modules, you will choose 2 of them. So therefore, you will be doing this one or a combination of any of these 3 modules to actually reach the 180 credits that you will need to obtain the grade of MSc and neuroscience. So the last 7 weeks block is of 2 optional modules, where you will learn how these 3 methodologies that you have learned and practiced during the core modules advance our understanding of neurodevelopmental disorders and disorders appearing in the adult age, like schizophrenia or bipolar disorder. Or optionally, you will also learn about the advances in our understanding of neurodegenerative diseases, and you will hear more about this from my colleague Rebecca Sims later on. Once completed all the taught component of our MSc, and from weeks 34 to 50, you will develop a research project. So you will be allocated to a group of supervisors, and you will develop a research project that will end up in submitting a dissertation in the form of an article submission. How your weeks will look like if you were to engage in this MSc Neuroscience? Well, this is the 4 first weeks, which will be a little bit more intense because this corresponds to the fundamentals of module. And in blue, you see the lectures or in-class teaching. In purple, you have seminar, journal clubs, and formative assessments. And in yellow, you have the practical sessions that are always 2 hours. And then you have assessments. You have formative assessments after the end of each half of this block. But then you have a summative assessment as well at the end of the half of the content of the block, at the end of the other half, and then a final summative assessment of all the teaching that you have received throughout this 4 weeks block. And then the rest of the blocks, of the 7-week blocks, will look like this. Again, in yellow you have in-class teaching, in blue, in yellow you have practical sessions, and in purple you have the seminars and journal clubs, along with formative assessments and summative assessments halfway through the modules, and another one at the end of the module. Importantly, during the weeks where there is assessments, there is no teaching happening, and there is never teaching happening on Wednesdays. All these green spaces correspond to self-teaching or time that you will be not supervised by lecturers or your supervisors, but that you would dedicate to self-learning and reading and preparing for the module. So that's how your weeks will look like. That's in a nutshell what the program is all about. But you have this QR code at the top right of this slide. If you follow that QR code, it will take you to the web page where all the information about this program is, and our contact details. In case you have any questions that you want to resolve, you are more than welcome to contact me, and I'll answer any questions that you might have. So I'm going to stop sharing my screen now, and I'm going to pass the word to my colleague, Dr. Rebecca Sims, that will take you and will give you some taster of the type of knowledge that you will learn in one of the modules of our program. Rebecca, whenever you want. Thank you.
Thank you very much. So give me 1 second and I will hopefully get this up and running. So hopefully you can see that. So yes, my name is Rebecca Sims. I'm a senior research fellow working at the Dementia Research Institute at Cardiff. And I'm just going to show this slide that Xavier's already showed you, but essentially to tell you where what I'm going to talk to you about is going to fit within the course. So I'm one of the module leads for the Advances in Understanding Neurodegenerative Disease module. So everything I talk about is what you're going to experience and the people that you're going to be meeting as part of that module. So I thought I'd start by just going over what the Dementia Research Institute is. So it's actually a UK-wide institute that encompasses 9 centres. It's got over 1,000 scientists and is classed as one national institute, even though we have 6 separate centres. Originally 6, sorry, now 9 separate centres. So Cardiff is one of these 9 centres, and the main focus at Cardiff is looking at exploring the function of genes that are found to associate with dementia. We primarily work on 3 dementias: Alzheimer's, Parkinson's and Huntington's disease, although that is not exclusive, and we do a bit of work on MND and other dementias as well. So first of all, I was going to talk to you about my focus of work, which is actually the gene discovery element. So where we find genes that associate with dementias. And this schematic is called a circular Manhattan plot. So it shows you the association peaks for genome-wide association study. And this is one of the most recent ones that was published last year. And we're also doing quite a lot of work, and this is a picture of Professor Valentina Escott-Price. So quite a lot of work including artificial intelligence and machine learning models in our discovery work to try and increase the genes that we're identifying for disease. So why do we look for genes? Why are we important? Why are we interested? So drugs are actually much more likely to succeed in disease if there is a genetic underpinning for them. So if there's a genetic element that is leading to their mechanism of action. So once we've identified genes, we can potentially identify coding variants within genes. So this is a paper we published nearly 10 years ago now, which have identified rare coding variant changes in 3 genes. And the work of the UK DRI since then has gone on to look at these gene changes and try and understand what they do, how they affect disease mechanisms, and therefore lead to the development of disease. So one example is PLCG2, which we've done some work looking at protein modelling. So this picture shows you the changes in the protein based on the risk variant and how that changes the protein conformation and therefore affects its function. We've also been doing a lot in animal models. So this is a picture of Gaynor Smith, who is one of the other co-lead of the module, and her work focuses on using the fruit fly in Alzheimer's disease predominantly, but again, other dementias. And one of the Alzheimer's risk genes, WOX, her group has been looking at how that impacts amyloid pathology through the fruit flies. And this picture on the right shows you the phenotypes we can see in those fruit flies based in neurodegenerative disease. So on the right, you have a tube with healthy flies where they have very good locomotive skills. They move around the tube. They tend to migrate to the top of the tube. In fruit flies that have been given an amyloid pathology, so this is where A beta is overexpressed in these fly models, you can see that they have a reduction in movement and they actually don't go where they're supposed to, essentially. So what a lot of Gainer's work does is look at trying to rescuing the Alzheimer's phenotypes by gene editing within these models. Now I'm not moving forward, sorry. There we go. So something else we look at with the genetics, so we find genes and we can look at specific single gene mechanisms, but we know that Alzheimer's disease in particular is a very polygenic disease, so it has over 100 genes that are now linked to disease. And what we can do with this is look at your cumulative risk instead of looking at a single gene, where we essentially add up all of the genetic risk elements that a person has. So we've been working on this for 11 years this year. 2015, this paper was published initially. So we've been looking at how we can predict who will go on to develop disease. One thing we've done from this is actually develop stem cell models. So rather than looking at a single gene in a stem cell model, we're looking at the cumulative risk within a stem cell model. So we have Alzheimer's people with very high risk and people that are not with Alzheimer's, with very low risk for Alzheimer's, and looking at what differences these genetics mean in cell phenotype assays. We also have been interested in looking at these in pathways. So rather than looking at a whole genetic risk for Alzheimer's, looking at which pathways are involved and then the risk within those pathways. And we've seen some evidence then from imaging studies using these pathways to show that people, even young people, have changes in their brain structure based on their polygenic pathway risk. And we've also been doing this with general polygenic risk, looking at hippocampal imaging work. So I mentioned biology. So the genetics, when it first came out in 2009, it showed us a lot of new biology that we didn't know was important in Alzheimer's disease. We'd known for a long time, thanks to the deposits of amyloid and tau in the brain, that the amyloid beta pathway and the tau pathway were very important in Alzheimer's disease. But we didn't know a lot about other pathways that were involved in disease. And these actually came as a bit of a surprise when we first did the initial genome-wide association studies. Then one that's come out very strongly is imaging. From the arrays was immunity. So the immune system actually plays a really significant role in the onset of Alzheimer's disease. And this was quite surprising because although there's a lot of evidence for the immune response in Alzheimer's disease, it was always thought to be secondary to disease. So the disease caused an immune response rather than the immune system helping in disease development. So this is a picture of Viola Zelek and Paul Morgan, Viola will also be teaching on the module, and the work that they've been doing looking at the complement system. So this is a part of the innate immune response, and they've been looking at the complement system pathway in human AD brains, as well as looking for biomarkers for disease. So essentially looking to see if we can use complement proteins to predict who is going to develop Alzheimer's disease and look at using this as a diagnostic. And their work has led to some translational work with industry. So looking at developing big molecules and small molecules that actually can cross the blood-brain barrier and actually target some of these disease processes. Another person you'll meet on the module is Caleb Weber. So his work is in Parkinson's disease rather than Alzheimer's. We sort of shift into the Parkinson's disease field in this slide. And what Caleb's group does is work with big data. So they look at transcriptomics. So this is RNA sequencing largely, but they look at single cell RNA sequencing across the brain, and then looking at creating what is called a multi-omics brain atlas. So looking at different cell types in different regions of the brain and mapping which genes and which cell types are important in those areas. And you get some very nice, pretty pictures, as you can see here, of regions of the brain and the types of cells that are seen in those regions of the brain. And then you can also map the proteins that are interacting and at working those areas. So it's a very specialized, a very niche analysis where you're actually looking at single cells and single genes within very specific regions rather than looking at a group of genes and a group of cells across the whole brain. So it's telling us a lot more about the specifics of these areas and the specifics of the cell types in these areas. Another work in Parkinson's disease is that with Dane Biaco-Kelly's group. He looks very much at the synapse in Parkinson's disease and aging as a whole, and looking at what changes happen at the synapse during general aging and during Parkinson's disease. And he will be one of the people that teaches on this module as well. And then we're moving to Huntington's disease. So Tom Massey is actually the first person you'll meet on the module. His work has very much been looking at the Huntington protein. So Huntington's disease is caused by a short tandem repeat, a CAG length in the Huntington gene. And we know that as the number of CAG repeats increases, the age of onset of disease increases. So this is what we would term a monogenic disease. However, there is a lot of variation, and you can see from this diagram here, there is a lot of variation in age onset for Huntington's disease, which is not explained by the CAG repeat. So the red line is the average age onset based on your CAG repeat length. However, you can see by the black dots that people get it a lot earlier or a lot later than they should. And that is because we see some additional genetic influence looking at disease modification, so age onset modification in this case. And a lot of Tom and others' work has been looking at the role of DNA damage and repair in this modifying role. And we also then taken that forward is Vincent Dion. So he's been translating some of this work in the lab, looking at CAG repeat lengths and seeing if actually we can reduce or eradicate disease based on reducing the number of CAG repeats once getting into the normal non-pathogenic range. And this work has been the sort of basis for the Huntington's gene editing or gene therapy trials that Xavier mentioned and Anne will be talking to you about next. So that's a great point for me to finish and hand over to Anne for the next section. Oh, you'll just stop sharing. So Anne, over to you.
Okay, so for some reason I can't seem to switch my camera on. Apologies for that. Let me just get my presentation up. Okay, can you see that okay?
Yes, we can see it. Thank you.
And it's in presentation mode. Okay, so I'm going to pick up from where Dr. Sims left off on the theme of advanced therapies, and by that I mean gene and cell therapies for Huntington's disease research, because both gene and cell therapies and Huntington's disease research are areas where we've got considerable depth and breadth in Cardiff, with work spanning the basic genetic and molecular biology right through to clinical application. So I'm going to focus on the development of a cell therapy for Huntington's as an example of how research in Cardiff spans a wide range of expertise and different methodological approaches, and in particular crossing disease modelling, human imaging, and the range of techniques needed to develop and test new therapeutics. So Huntington's is a devastating inherited neurodegenerative condition, but what you need to know for this talk is that the main area of degeneration is in the striatum, which is the deep brain area you can see on this diagram, and it's the principal nucleus of the basal ganglia. So the reason that the striatum degenerates in Huntington's disease is the progressive death of a specific sort of neuron called the medium spiny projection neuron, which I may sometimes refer to as MSN. And so the idea of cell replacement therapy is that we aim to transplant new cells into the brain to replace the function of those that have been lost. And to do that, these cells have to survive and reconnect the neural circuits. So this is work that's been going on over a couple of decades in Cardiff and has involved a number of groups within Cardiff University and collaborators elsewhere. And the starting point for this was to develop a robust and reliable differentiation protocol so that we could take stem cells right through to being medium spiny progenitors. And so to do this, we had to apply developmental principles to drive stem cells from a fully pluripotent state right through progressively more differentiated states to an MSN progenitor phenotype. So MSN, medium spiny neuron progenitors, are amenable to transplantation, so they'll survive the transplant process, but they're specified to develop into mature medium spiny neurons over time in the brain. And so we've used various ways to confirm that these cells are properly differentiated to medium spiny progenitor subtypes. And so, for example, you can see on the left, we've used immunohistochemistry, where we stick the cells down in a dish and label with antibodies to see if they express proteins that are typical of medium spiny neurons. And in the middle, you can see that we used PCR to check whether the patterns of genes being expressed or repressed were as we would expect for these particular neuronal subtypes. And on the right, you can see a more unbiased approach, such as using single-cell RNA sequencing to look at all the genes being expressed in individual cells so that we can understand exactly what cells are contained in our cell product. But as well as characterizing our cells in a culture dish, we needed to know how they behave when they're being transplanted into the living brain and you know, so that they've had many weeks to grow and develop. So to do this, we need to transplant them into animal models of disease. And there are a number of groups within the university who work with various sorts of animal models. And in the Cambridge Brain Repair Group, we have a range of animal disease models and also small animal surgical facilities, which mean that we can undertake neural transplant experiments. And you can see an example here on the right of cells transplanted into a rat model of Huntington's disease. And these are brain slices that have been stained for markers of medium spiny neurons, such as DARP32. And you can see the graphs here packed with DARP32 expressing cells. So as well as being able to visualize the survival and differentiation of transplanted cells, we've got a whole host of techniques that are available to us to explore how they're wiring up within the brain and how they're functioning. So here on the left, you can see that we've simply used a human-specific cytoplasmic antibody in brain slices to allow us to see where the human cells have projected to in the rodent brain. But there are also other more sophisticated and more powerful techniques. So, for example, on the right, we've used a modified rabies virus that can travel across a single synapse. And this allows us to trace cells that are connected to the graft. So in other words, cells coming from the host brain into the graft. And this allowed us to determine that several areas of the host brain were sending projections to the brain and making functional synapses. So in other words, it looks like the grafts are connecting up. And another way we can assess the grafts is to assess whether the grafted cells can fire action potentials and look at whether they can re-establish normal electrophysiological profiles. And this slide shows some slice electrophysiology that we did with collaborators in Cardiff and elsewhere. And here we made slices through the area of the brain that contained the graft. And we were able to do recordings and show that the grafted cells had established electrical connections with the remaining host striatal tissue. But finally, the most persuasive evidence is to show that the grafts can improve functional deficits in whole animals. And we're fortunate in Cardiff to have the expertise and the equipment to undertake a range of behavioural tests, and some are illustrated here. So, for example, tests of rodent rearing and paw reaching, and automated testing boxes which can be programmed to assess a range of behavioural functions. And using these sorts of approaches, we were able to show that our grafted cells do have a positive effect on cognition and movement. And we're now working to take this technology through to a first-in-man clinical trial. And taking the therapy right through to a first-in-man clinical trial is possible here in Cardiff because we have the setup to do human studies. And we have a number of institutes. For example, we have Kubrick with world-beating imaging technology that allows us to understand the brain in these disorders, and also to see what happens in response to therapeutics like our cell therapy product. And we also have the Advanced Neurotherapeutics Centre, which has surgical facilities and access to on-site brain imaging. And we hope to use these facilities to deliver our cell therapy and other new therapies that are being developed in Cardiff. And we're confident that we can do this because we've already used these facilities to deliver gene therapies directly to the brain for 3 different neurological diseases. But I'm just going to tell you very briefly about one of these, which is the uniQure AMT-130 gene therapy. And this is a therapy that was delivered directly to the brain. And Cardiff was one of only 2 such centres in Europe that had the capability to deliver this therapeutic. And this caused some excitement back in September of last year, when the company uniQure announced its top-line results, which showed that this gene therapy provided the first demonstration of disease slowing in Huntington's disease. So we were very proud in Cardiff to have played an important part in this groundbreaking research. So I hope I've convinced you that within Cardiff we have a range of techniques that really cross the translational spectrum, that we work in a cross-disciplinary way, and that the work has real-world impact.
Thank you very much, Anne. I'm convinced, so I hope everyone else is convinced as well. What I'm going to do now is I'm going to stop the recording and we'll open the floor to questions. Thank you very much.